Assalamu alaikum everyone, welcome to my channel. Today we will start our fourth chapter of biology, that is cells and tissues. As you know, everything is made up of cell. It is the basic structural as well as functional unit of life. All organisms are composed of cells. Some are composed of single cell and some, like humans, are composed of many cells. For example, the wings of a butterfly is a thin sheet of cells, and so is the shiny layer of our eyes. As well as the meat we eat is composed of cells, and its contents soon become parts of our cells. Thus, everything is made up of cells. In this chapter, we will discuss microscopy and the emergence of cell theory first. For this, we must know what is microscopy. So, microscopy is the use of microscope. The simple definition of microscopy is that the use of microscope is known as microscopy. Compound microscope. The first compound microscope was developed by Zacharias Janssen in Holland in 1595. It was simply a tube with lenses at each end and its magnification ranged from 3x to 9x. पहला कंपाउंड माइक्रोस्कोप जो है वो ज़कैरियस जेंसन ने हॉलैंड में 1595 में डिस्कवर किया था. इट वॉज composed of only a simple tube lenses at each end and its range was 3x to 9x. There are two important terms that are used in microscopy. These are magnification and resolving power. Magnification is the increase in the apparent size of an object, and it is an important factor of microscopy. Resolving power or resolution is the measure of the clarity of an image. It is the minimum distance at which two objects can be seen as separate objects. And the resolution of human eye is at least 0.1 mm apart. In microscopy, we will discuss what are light microscope and electronic microscope. There are two types of microscope, light microscope, also short form is LM, and electron microscope, short form is EM. These microscopes are used in microscopy. First of all, light microscope. All light microscope works by passing visible light through a specimen. लाइट माइक्रोस्कोप में स्पेसिमेन के अंदर से जो है वो लाइट को गुजारा जाता है. इट यूजेस टू ग्लास लेंसेज. वन लेंस प्रोड्यूस इन ए लार्ज इमेज of the specimen and the second lens magnifies the image and project it into viewer's eye or on the photographic film. Basically, a photograph taken through a microscope is called a micrograph. Means that the work that is done by a microscope is called microscopy and a photograph taken through a microscope is called micrograph. There is the evolutionary image of light microscope in your book. Let us discuss the function of light microscope. A light microscope can magnify objects only about 1500 times without causing blurriness. For example, its magnification is 1500x. Its resolving power is 0.2 micrometer and 1 micrometer is equal to 1 per 1000 millimeter. In other words, the light microscope cannot resolve or distinguish objects smaller than 0.2 micrometer. It is about the size of the smallest bacterium. The image of bacterium can be magnified many times, but the light microscope cannot show the details of its internal structure. Next is electron microscope. Electron microscope is the most advanced form of microscope. In electron microscope, object and lens are placed in a vacuum chamber and a beam of electrons is passed through object. Electrons pass through or are reflected from object and make image. जो इलेक्ट्रॉन्स हैं वो इमेज के अंदर ऑब्जेक्ट के अंदर से गुजरते हैं या उसको रिफ्लेक्ट करते हैं, तब जाके एक इमेज बनती है। इलेक्ट्रोमैग्नेटिक lenses enlarge and focus the image onto a screen or photographic film. Electron microscope has much higher resolving power than light microscope. The most modern electron microscope can distinguish objects as small as 0.2 nanometer, and 1 nanometer is equal to 1 per 10 lakh millimeter. It is a 1,000-fold improvement over light microscope. Electron microscope can magnify objects about 250,000 times. Under special condition, electron microscope can detect individual atoms, cells, organelles, and even molecules like DNA and protein are much larger than single atoms. Types of electron microscope. There are two types of electron microscope that scientists use, which are transmission electron microscope TEM and scanning electron microscope SEM. In transmission electron microscope, electrons are transmitted through the specimen. Transmission electron microscope is used to study the internal structure of cell. Electron microscope has revolutionized the study of cells and organelles, but it cannot be used to study life process because the specimen must be held in a vacuum chamber. For example, all air must be removed. To study the life process, for example, movement of amoeba, light microscope is better. And the function of scanning electron microscope is that it is used to study the structure of cell surface. This is it for today's video.